Hello and welcome. For this video we are looking at the idea of distribution of living things, and by distribution we mean just how they are spread around in their either habitat or their environment. And there are a few factors that will have an impact on how they are distributed. So those factors are temperature, the amount of light, the availability of water, the availability of nutrients, and the availability of oxygen and carbon dioxide. All these things will have an impact on how these living things are distributed. We are really going to focus more on plants for this video, and there are one or two other things that might be involved. For example, pollution might have an impact, and other things like predators or new competition or something like that. So new competitors being introduced. But anyway, so we're going to look at how we make estimation of the distribution of living things, and we're going to focus on two methods, and those methods are random sampling with quadrats and sampling along a transect or along a transect line. And the first one we're going to look at is random sampling with quadrats. Now here is a rather badly drawn green area, or a field if you like. Over this side I've got some trees. This is supposed to be a stream flowing through. This is a little pond, and this is a pathway that's going all around this field. Now you might want to do an estimate of the number of different living things there are in this field, and you might, for example, want to look at the distribution of dandelions and daisies and how they are distributed around this field. Now the method that we're going to talk about first is this idea of random sampling with quadrats, and the first thing we need is our quadrat, and a quadrat looks a little something like this, usually half a meter by half a meter, but it can have different sizes, and sometimes it's subdivided with string into the smaller squares, as you can see there. And you would use this quadrat to help you measure the distribution of these two different species of plant. So what you might want to do is start off in one corner, get your quadrat, and maybe place it in a certain area. Now the problem with you deciding where to place it is that it's not going to be very random. It might not be very representative. You might think, well, there's a lot of dandelions in here, so let me chuck the quadrat over here, so I've got something to record. So it's very important that those factors don't have an influence, and you would try and make it or throw it or place it randomly as possible. Now one way you can do that, as I say, is to just throw it. You could close your eyes or throw it over your head backwards, or you could actually make a grid, produce a grid, and pick out random spots from the grid in order to take your readings. But the point is, it must be done randomly. You can't have any kind of system when we're doing random sampling with quadrats. So what you would then do is you'd get your quadrat, place it at your random points, and take a series of readings, and you can then measure the number of dandelions and daisies for each particular quadrat, and you might get a table of results that looks something like this. Okay, so here we have the quadrat number. In this case it's been thrown 8 times, or it's been placed 8 times. We've got number of daisies for each quadrat, and we've got number of dandelions as well. And there's some data analysis you can do with these numbers, and there are 3 ones that we want to focus on and concentrate on today. The first one is called the mean, and if you're quite good at maths, I'm sure you've come across this in maths before, but we've got mean, median, and mode. The first one is mean, and the way you work out the mean is you look at the total of all the values, and you divide it by, I've got the dividing line there, you divide it by the number of values that you have. So if we were looking at the mean number of daisies, we would look at the total, and I think the total in this case is 48. Okay, so if I add all these up, I get 48, and I divide by the number of values, and I've got 1, 2, 3, well, I've got 8 values there. So the mean number of daisies is 6. Okay, so we've got 6 per quadrat, and that gives you an idea of how these daisies are distributed across this field. For the number of dandelions, you would do the same thing. So I think in this case I've got a total of 8 divided by 8, so I've got a mean of 1 dandelion per quadrat. Okay, so that's working out the mean. Now if you want to work out the median, well, this is the middle value, and for the middle value, you might remember from your maths, is the first thing you have to do is to put the numbers in order. So the lowest number here is 3 for the daisies, I've got 3, then I've got 5, then I've got 1, 2 6s, then 4 7s. And the median is just the middle value, so you'd go along and find the middle value. I've actually got 2 middle values here, and those are 6 and 7, and for the median for this one you just take the midpoint between those two, so it'd actually be 6.5, so that's the median. I'm guessing they wouldn't actually give you an even number in the exam, they'd probably give you an odd number, so you'd actually have one number in the middle there, but having said that, you never know, so good to know that if you've got 2 middle values, you just find the middle of those two. So the median for the daisies is 6.5, and the median for the dandelions, in the same way, you put them in order. So we've got 0, 0, and then 1, 2, 3, 4 ones, 1, 2, 3, 4, and a couple of 2s. Is that enough? 1, 2, 3, 4, 5, 6, 7, 8. Right, so there's the numbers put in order, and the middle value, again, I've got 2 numbers there, but they're both 1, so the median for this case is 1. And the mode, this is the third thing that we need to know, and the mode just means the value that occurs the most. I tend to remember it by saying mode, sounds a bit like most, so that's the meaning of mode. So for the daisies, the mode, most often occurring value is the 7, and for the dandelions, the most often occurring value is the 1. Okay, so the mode for the dandelions is 1, the mode for the number of daisies is 7, and you need to be able to work with or calculate these values and use those for a set of data that you might be given. The next thing I want to look at is how we would sample along a transect, and examples of things that you might look for using a transect is you might look at grass under a tree. So I've tried to show 2 different types of grass. Got these long, slightly darker ones and the shorter, brighter ones, and you can see they're distributed slightly differently along the tree there, under the tree there. You might look at these things called lichens, which you might have seen growing on trees or sometimes on walls of houses or brick walls that you might see. You might look at moss growing on trees or walls, and there's a whole different variety of things you can look at using this idea of sampling along a transect. And the way we do it is like this. So you start off with the thing that you're measuring. So let's say we're measuring the grass under the tree. So you get a tape measure, or you could use a marked rope or a string that might be marked every meter. You place that along the place where you are examining, and you would then place your quadrat. So I just need my quadrat. There it is, and you place it along the first point there. Now this is standing up. Obviously you wouldn't have it standing up, it'd be lying flat there, but you place it along your first point there, measure the type of living thing that you're looking at, and then move it along. Record the presence of each type of plant, and then move it at the next point. So you'd move it a meter along, and you'd measure that on your tape measure or your marked rope, and you'd then do the same thing again, measuring the different type of each plant, and then you'd go along with your quadrat measuring the different species that you're looking at, and again record it in a table similar to what you saw on the previous page, and then you can do your analysis. The other thing is you would probably want to do it a couple more times, repeat at least 3 or more times, 3 to 5 times maybe, and that's in order to get what we call reproducible or reliable results. And I'll talk more about that in a sec. So the other thing I just wanted to quickly go over, I've seen something like this in a past exam paper. We're looking at, again, this is sampling using a transect, but it's across a stream, or this might be a pond actually. We've got different types of plant species, and we've recorded it in a slightly different way. We've recorded it like this. So we've got distance along our transect in meters along the bottom, and then we've got the plants listed on the sides, and these bars just represent whether the plant is present or not. So for plant A, you can see it's present at this distance here, so you can see we're talking about this long sort of grass type of plant there. For plant B, we can see it appears just under a meter or half a meter onwards, and that's that plant there. We've got a large amount of plant C along this middle section here, and that's the one that's under the water. This plant here only seems to appear on this side, and if you look at plant B, it appears on both sides, so it's here and here, and similarly for the grass, it appears on both sides. And so you can look at this, a table that looks a bit like this, and you can see what the distribution of the different plants are just by comparing the two diagrams like so. So once you've got your data for all these different possible methods, what you would want to do then is maybe go back and actually link it back to what we were saying at the beginning. You might link it to the different types of temperature or amount of light, or all these different factors that might vary in the two areas or in the areas that you are examining. Now, the last thing that's really quite important to mention is that you have to try and make your results or your recordings valid and reproducible. So you can imagine that here, for this experiment here, if we only took one reading, say we took this reading number 7, that was the only one we took, we would assume that there are no dandelions in the field because we only took that one reading, and that would not be valid because there are actually dandelions in the field, but that one reading didn't show it. So we would take readings that are valid and reproducible, and this is usually linked to sample size. Okay, so you want to make your sample large enough so that you can make it valid, and you want to see that it is reproducible as well. So if somebody else came along and did the same thing, they would get very, very similar or close results to yours. Okay, so that's just basically an overview of our distribution of living things. We've tended to focus on plants, which is what we tend to do when we're looking at sampling with quadrats and sampling along a transect. But that's it, that's me done for this video. Thanks for watching, and I'll see you again soon.